So what we talked about real quickly at the end of last class, I believe, was up here in our pyruvate dehydrogenase, right? And we had this lovely figure that we kind of stepped through. The pyruvate ultimately getting converted out into, whoops, circled too much there. Getting converted out into acetyl-CoA. And that step, pyruvate dehydrogenase, that's our big step. Pyruvate to acetyl-CoA. That's all we're really trying to do. The rest of the stuff that's happening in there is to facilitate it, okay, to allow it to occur. Right? And because of a lot of those big changes, there's a lot of stuff involved. Okay, so the one that I want to look at in particular is our thiamine pyrophosphate, which we already saw in glycolysis in a different dehydrogenase enzyme. I believe it was a dehydrogenase. Anybody remember? I think it was a dehydrogenase. So the exact details of what all these different little pieces are and how they work, not super critical. What I want you to get out of this is, number one, our thiamine pyrophosphate, because we're going to look at that in closer detail. We're starting with pyruvate. We're using the dehydrogenase enzyme. Okay? We're making acetyl-CoA, and we are making NADH. Okay? So pretty much 5 big things out of this. And I'm going to add one more. The NADH is actually getting made by FAD. So we're using FAD as kind of the intermediary to allow for the oxidation to refresh the enzyme. FAD then dumps electrons onto NAD.
And in that case, the FAD is attached somewhere.
It's,
the NAD is free floating. FAD
yep.
takes, the NAD comes by and takes that. And
Yeah, the FAD is pretty well buried inside the enzyme. Okay. And FAD is going to come up again, being buried inside the enzyme in our electron transport chain. So at this point, we've pretty much just said this stuff is happening. We didn't say where it's happening. Right? As soon as we move into the electron transport chain, that's when we'll start saying, oh, remember that enzyme? Here it is. It's located in this place in the cell, and this is why we can use these electron transports as we can. Okay? So thiamine is cool because we form a carbene. Whoops, sorry. Okay. From OChem, a carbene is this structure. We have an sp2 hybridized carbon, so 3 electron groups, okay, which means we are missing an octet on that carbon, which means there's an empty orbital. That empty orbital can accept electrons. So our carbene can act as an electron acceptor, but then it also has a lone pair of electrons, which means it can act as an electron donor. So this is a really neat atom in this case because it's doing 2 different things. Okay? So that's kind of fascinating and cool. When we look at it in organic chemistry, it's not easy to go through and make. We've got some pretty strict conditions to force that to happen. So we would assume that this molecule, there's no way in hell it could possibly happen in biochem because we have some very specific and nasty conditions to force the carbene to form in organic. It turns out biochem, we do end up forming it as well. The carbon in our thiamine ends up replicating the carbene reactivity. That hydrogen is relatively acidic. So that means I can get rid of the hydrogen, in which case I now have a lone pair of electrons and a negative charge. Unfortunately, this figure was not the greatest, and there's a pretty big typo in it. What's that typo? There's a subtle hint. Yeah, our nitrogen is actually positively charged. Does the nitrogen want to become positive? No, so these electrons can get pulled away from the carbon.
So is there a strong base that's going to pull that hydrogen off? Or is
so that hydrogen is relatively acidic because of stabilization within the structure. We've got 2 electronegative atoms, so we have kind of an inductive effect, making that hydrogen pretty acidic. Okay. And that's one of the reasons why we can pull that off easily.
Oh, so you don't need that strong.
We don't need that strong of a base. Okay. relative to biochem, it's still pretty strong, but our amino acids have enough of a base strength that they could pull that off. Okay. So that carbon can become the equivalent of our carbene, okay, which becomes useful when we use it in our dehydrogenase. So if we look at our very first step, what happened is we've removed the removed the hydrogen. We now have our active negative charge. You'll notice we aren't showing it as the carbene form with the electrons going to the nitrogen. Okay? And that's because the full-on carbene form is not particularly stable. So we need something to kind of satisfy it, running at octet rules, all that kind of fun stuff from Gen Chem and Lewis structures and first semester OChem. Our pyruvate molecule has 2 carbonyls. Okay? So our carbene will act first as an electron donor. So now the question is, where is the electron acceptor? So we need a positively charged carbon. Where are there positively charged carbons in pyruvate? Okay, we have partial positives at both of those carbonyl carbons. So why does it selectively go to the ketone and not to the acid? The answer to all things, there's resonance over here, which stabilizes the positive carbonyl from our acid, which minimizes its reactivity. The ketone is going to be the one that's more reactive. Okay? So we end up saying the attack as shown. This then links the pyruvate to the structure. Okay? Nitrogen, being positively charged, wants its electrons. Okay? So let's focus there. It says, finally, that nitrogen is finally happy, it's neutral, I'm going to take my freaking electrons back, this is annoying. So it takes the electrons. What happens then to our carbon? It's now positively charged. This is where our carbon can now act kind of as an electron acceptor. Okay? As that positive charge, it can accept electrons, but it needs to get the electrons from somewhere. Okay? And this is where this enzyme is really helping us out. What do we have right here? Yes, we have extra electrons in that negative charge. We have an oxygen. We have a carbon, and we have an oxygen, known as CO2. Okay? So the electrons from our oxygen can come down to form the double bond to give us CO2, but then that central carbon, our purple carbon, has too many electrons, so it has to eject electrons. Typically, ejecting those electrons, we'd eject them onto that carbon. Okay? But then we'd have a negatively charged carbon. Can we have a negatively charged carbon? No, that's not easy to go through and do, particularly in biochem. So that's why pyruvate does not do this on its own. Something else needs to get in there to help polarize to allow for the stabilization of that negative charge. What is that something else? Those electrons, instead of going to the carbon, can go to form a double bond between the carbon of pyruvate and the carbon of our thiamine. Okay? The result is we've now formed our double bond in that system. Right? And we've lost our CO2. So that's our big, big step. We've now ejected the CO2. And Jimmy, you should be used to it by now, but I'm going to call you out again. All right. You're welcome. Okay, I'm going to make that nitrogen and oxygen here temporarily. What do we have? We have not only just alpha, and it's not necessarily alpha beta unsaturated, it's that beta keto acid. Okay. At the beta position to an acid functional group, I have the equivalent of a ketone. Okay. If you go back and check OChem, second semester OChem, towards the end when you're looking in your carbonyl chapter, that was a really fancy structure, because what that structure could do under the proper conditions, it could do a decarboxylation. Well, what did thiamine just facilitate? Decarboxylation. Okay? It's not technically the same thing, okay? Because if we go back and look at this, we're looking at a nitrogen at that position instead of the oxygen. Okay? If we go through and look at pyruvate, why does pyruvate not do that? Well, at our beta position to our acid, so there's our acid, alpha, beta, there's no carbonyl. So we don't have a way to polarize and allow for the electrons to shift out. Ultimately, what's happening what functional groups are we looking at equivalently here? In the red we have an enol, and in the blue we have the equivalent of an enol that we refer to as the enamine. Okay, and we have to. It's not an enol or an enolate because both of those involve oxygen. Here we're bringing in a nitrogen, so we have to change the name. Okay, but it's the same equivalent chemistry. That whole resonance with the double bond, keto, alcohol, alkene forms, all of those are going through the same processing. Okay? The rest of this situation is pretty much coming up with a way to now kick the thiamine back off. Okay? So if we go through the rest of our process, our hydrogen becomes acidic, because if the hydrogen's acidic, we can go through and reform our enol-enamine system. Okay. This can be helpful because we can now generate a nucleophile in our acetyl group, or the equivalent of our acetyl group, that can then be picked up by our lipoic acid. That then can help shuttle it to eventually kicking the thiamine back out on its own, which they're showing as TPP. We now have our acetyl group connected to our lipoic acid. Okay. Through a next sequence of steps, coenzyme A can come in, facilitate, and do our full transfer to get us coenzyme A with an acetyl group. The big parts of this that I want you to be familiar with are the parts that we've colored over at the top. Everything there kind of after, not as big of a deal because we're bringing in the next step in that cycle. Okay. So this first part was pretty much all our TPP doing what it needs to for our decarboxylation. Okay. And that gets us all the way up to this step, which now brings in our lipoic acid, okay, which we then have to go through some fun kind of intermediates, our tetrahedral intermediates, where we can eventually kick out our thiamine. That's our TPP. So we're refreshing our thiamine molecule. Okay. But then we need to get that piece out to coenzyme A, which is the next sequence. Okay. Any ideas why we have to go to lipoic acid and then to coenzyme A? Why not just bring coenzyme A right out of the gates? I'm guessing at this point too, by the way.
Check and balance.
It can be a check and balance. How reactive was thiamine? It's a carbene. Were carbenes super stable? No, carbenes were super reactive. Where are we going to want to put a super reactive thing? It's a super reactive thing that humans have control of. Oh, uranium. Okay, what do we do with uranium? That's freely available on the open market, right?
Stored away.
Okay, we hide these really high energy particles because we don't want them to get out and react. This enzyme is massively large. It is now trying to hide and bury our reactive intermediates so that it doesn't react with anything else, say like DNA. Okay, that could be a bit of a problem. Okay, so we're trying to bury those reactive structures inside something. Well, if it's buried on the inside, can we get mobile molecules from the outside in? That becomes increasingly difficult. So what we'll end up doing is kind of burying our chemistry and slowly working its way back out to the surface. Okay? That's likely what's happening in here. is that we've buried the major chemistry into the middle of the structure, and as we process our way through, we're slowly moving that piece further and further to the outside of the enzyme, where it can now be more accessible by our, say, coenzyme A, so that it can then react and easily transport out. That way our chemistry stays relatively contained. Yes?
It seems like structure 3 and structure 5 are the same structure
1, 2, 3, and
with your amine and your enol.
5.
It's like you got this random acid-base thing going on with 4, and then it just looks like it just undoes it. Like, nothing really
Yes,
that changed.
you're right. So let me clear everything there. You're referencing structure 3 and structure 5. If we do take a look at those structures, are they the same? Yeah, they are. Okay. So what's going on? Why are we going through to structure 4? What's the big deal there? Whoops, that should be a 4.
Are we just saying that that's also reactive, so you get one of these two things might be happening? Or just...
Which is more stable? Okay, your enol equivalent or the keto equivalent?
Oh, I guess our keto equivalent.
Your keto equivalents tend to be more stable, so more than likely you're shuttling through step 4 as a stabilization feature. Okay? This can give us enough time to delay until the lipoic acid comes back around to facilitate the reaction. So we're probably seeing in that sequence, this is an acid-base step, and that next one is an acid-base step. Those are probably happening very, very rapidly in comparison to everything else, but they're there to kind of stabilize our intermediates.
Are we going to hang out in step 4 until that lipoic acid
Yep,
comes around?
until lipoic acid is available. Almost as a check and balance. Kind of makes sense. So I think it's a neat reaction, doing a decarboxylation. It was a really hard one for me to figure out the first time because I kept looking at pyruvate saying there's no way in hell that can get rid of a carboxylate or a CO2. I don't understand what they're trying to tell me. Okay? And the reason why, pyruvate won't get rid of CO2. We need to add an extra piece in there, that's our thiamine. That's what's facilitating and allowing this reaction to occur. That's what sets us up with that beta keto acid. Okay. Now we can do the decarboxylation. Once we've done the decarboxylation, we effectively have our acetyl group. It is in kind of a weird intermediate structure, which is why we have to do all the other reactions to kind of get it shuttled back out to coenzyme A. Questions? Okay. So the big result out of that, actually one of the big results we can get out of that, if you want it done, protein does it. Okay? Take a look at your textbook, they kind of just say, oh yeah, this just happens. Okay? That's because they didn't want to explain the organic chemistry behind it. Okay? So when in doubt, your protein does it. So some kind of exam questions. Let's give this a pause here, give you a second. Officially, your best answer would be a nucleophile because we have two different species there. I would accept credit for an electrophile because we've talked about it acting as both. And if we look at the following reactions within it, it does accept electrons, but it's accepting electrons from a structure that's already attached to it, so it becomes difficult to label it as an electrophile or a nucleophile at that point. Okay. So both do work, nucleophile is better.
So in the previous slide then, it comes in at the first step?
If we look at that very first step off of our
The
previous
first
slide.
meeting at that first step, so that's when the donor part comes from?
This piece
And then
is
the
our
next
thiamine.
step, it's still there, and then now it turns into the acceptor.
So in the next step, if we subdivide or could draw out thiamine as a separate piece, okay, what is it acting as? It is accepting electrons. Okay, so it is kind of acting as an electron acceptor there. It becomes questionable because it's intramolecular within the molecule, and that's where we run into weird lines on defining what it's doing. Because by accepting electrons, it's also losing pieces, so it becomes a little bit odd to label it.
But in that initial step, though.
That initial step, it is very cleanly acting as a nucleophile. It is sharing electrons with an atom other than hydrogen. So, very cleanly a nucleophile there. And in the next step, it's an electrophile in quotes. Thiamine is an example of a naturally stable carbene. Okay? It's just recognizing the pieces that we've been talking about. Does the conversion of ADP to ATP require oxidation?
Yes.
No. So this, we'd have to go back to glycolysis.
Probably had oxidative phosphorylation or
Exactly.
something level.
Or
oxidation.
is the big word there.
But isn't it just the phosphate that's doing it?
So,
There's no oxygen there.
If we look at this process right here, we are generating ATP using a kinase. Did we do any oxidations?
So it just transferred the phosphate.
We just transferred the phosphate across. It does look like we've increased the number of bonds to oxygen, but this is where we have to be careful with oxidation and reduction. We've actually decreased the number of bonds, or sorry, we increased the number of bonds to hydrogen, but at the same time increased the number of bonds to oxygen. It's the easiest way to look at it, which then means increased oxygen, increased hydrogen means one is oxidation, one is reduction, the net result is nothing. Okay. The other thing you can look for on trying to decide that is NADH or FAD involved in this reaction. No. Those are your big carriers for oxidative phosphorylation. So if you see FAD or NADH or NAD, those are going to be your signifiers that, hey, oxidation is happening. Phosphorylation is, do we have a transfer of a phosphate? We have the transfer of phosphate, do we see NAD or FAD? No, so it can't be oxidative.
That reminds me of one of the homework questions. Basically said, which of all these steps in glycolysis is an oxidative step?
Yep.
And apparently only maybe step 6 or whatever that.
Yeah, I think that's right. We have to go back to where, there it is, where we use NAD. So if you don't see NAD or FAD, you're not doing oxidation or reduction. Okay, at least for the sake of this class. Those are your big electron carriers. There's other smaller ones, but those are your primary ones.
So in that case, the phosphate would not reduce or anything like that, it was that carbon on the upper left over there?
So yeah, the reaction that's happening here is that carbon.
That carbon is getting oxidized.
That carbon is getting
The
oxidized.
phosphate is just doing its thing with the phosphorylation, and it's not.
Yeah, so we are still phosphorylating here.
Yeah.
Okay, and it is oxidation. It's a little questionable to call it oxidative phosphorylation, but it is doing both technically. When we think about oxidative phosphorylation, it is typically in reference to the formation of ATP. Okay. And I'm not sure if that's exclusively the case. Okay, so we got to be a little bit careful on that terminology. When we're talking about oxidative phosphorylation, did oxidation happen and did phosphorylation happen? Okay. Yes, now it's what are we referencing within those, and that's where we have to be careful with the nomenclature within biochemistry. So we can get a quick hint, now we have to nail down the specifics of it, and in all honesty, I don't have those specifics nailed down.
So when it's just the phosphate being put on and taken off, then no reduction?
No oxidation or no reduction.
If anything else, like in this one, other stuff is happening,
Yeah.
then there it is.
So if we move to this one, okay, we are taking off a phosphate, so we are doing a phosphorylation. We are putting a phosphate onto ATP. So this absolutely fits our definition of phosphorylation. Did we do an oxidation or reduction reaction? No. So it's not oxidative phosphorylation because there's no oxidation or reduction, it's just phosphorylation. They've decided to add this extra term into it. It's not oxidative phosphorylation, but I want to call attention to it being phosphorylation. Why do we not just call it phosphorylation? I don't know.
Substrate level phosphorylation.
Substrate level phosphorylation is just saying it's not oxidation reduction, we're just putting a phosphate on or on. You mean phosphorylation? Yes. Yes, that is what I mean. But I'm going to say substrate level. Okay? So it's trying to call attention to the fact that it is not oxidative phosphorylation. It's just an extra emphasis. Okay. So if you're looking for oxidation or reduction, look for your key electron carriers. That has to be FAD or NAD. Okay. If you don't see them, ignore your oxidative phosphorylation, or oxidative part of it. And sorry, now we get scan back through. Where were we? So does the conversion from ADP to ATP require oxidation? We just looked at an example where it doesn't. Okay. Most of the cases where we are getting ATP does come from oxidative phosphorylation, because that redox reaction generates a lot of energy, and we can take advantage of that energy relatively easily to generate ATP with minimal heat transfer. The substrate level phosphorylation can have issues with heat transfer, so we tend to minimize those reactions. Okay. So we got a bunch of possible options in there. You notice QH2? We haven't talked about it yet. QH2 is another coenzyme, coenzyme Q. Maybe that rings any bells. Guess what it does? It's another electron carrier. Okay. Much less mobile than NADH. It's shifting around pretty much between two enzymes on the surface of the membrane. Doesn't go anywhere else. Very, 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 very local. Okay. It does still move, but not a lot. Okay. So we'll talk about that. ATP synthase is a little bit weird. To get ATP synthase to work, you have to do oxidation, but ATP synthase does not work by doing oxidation. Okay. So we'll talk about that when we get into the electron transport chain. Either way, your answer is no. Okay. The other ones, anybody know enolizable hydrogens?
Probably the only one with that enzyme.
Okay. There's our enol. The relationship to the ketone, what did we need? We need a hydrogen located at which position? Our alpha carbon. So which of those structures has a hydrogen at the alpha carbon? There's one. There's one. There's one. There's one. So that one's got a lot of enolizable hydrogens. Is there any enolizable hydrogens here? Nope. How about here? We don't have an alpha carbon, so we have to have that alpha carbon with hydrogens attached. In this case, the hydrogens, let's go with a different color, are not attached to an alpha carbon, they're attached directly to our parent carbon. And I think I just lost connection. No, I didn't. Okay. So those do not have enolizable hydrogens. Okay. So again, we didn't directly talk about it. I know we just directly talked about it, but we haven't directly talked about it in the past. I won't ask this kind of question. So recognize the patterns. That's going to be helpful. We haven't talked about citrate that word yet, because that is our very next step. We'll take acetyl-CoA and we will react that with oxaloacetate and water to make citrate. Okay? So we are making citrate. So if we are making something, good name might be synthase, because we are synthesizing, and it's an enzyme, hence the ACE. So synthesis enzyme, what are we synthesizing? Citrate. So citrate synthase. Okay. If we think about that, should it be controlled or affected by NADH? Okay. So if we look, there's our citrate synthase in that first line. Do we see NADH showing up in that? No. So what might be your first pass at that being affected by NADH? We might say not. What happens to citrate? Turns into isocitrate, which then turns into alpha-ketoglutarate. Alpha-ketoglutarate, to do that reaction, what do we produce? NADH. Okay. What if I have, and then NADH can later be used to do what? Make ATP. Okay. Do I want NADH to affect my citrate synthase? What happens if citrate synthase works? I take this acetyl group, put it onto oxaloacetate, I make citrate, and now citrate goes through and makes more NADH. If I already have tons of energy, what happens if I initiate that first step? I make more energy. But if I already have tons of energy, I don't want it. So should NADH have an effect there?
Yeah, negative feedback.
It should have effectively a negative feedback loop. So we would expect some kind of inhibition there, and inhibition. And then allosteric, okay, the specifics of that, not super big deal, but we would expect some kind of feedback inhibition there. Okay, and in this case, allosteric makes sense. Okay.
So
There is an effect. Okay. And one of the reasons, important things to acknowledge there is that acetyl-CoA has this really powerful acetyl group. Okay. That acetyl group can be used in a variety of things. Okay. We can use that acetyl group to reverse the reaction. Okay. We could start building glucose back up. Okay. Do fat stores. Or not glucose, but fat. Okay. Make some lipids. Store some energy. Okay. By now having that carbon chain react on something else. So if I have tons of energy, I don't want to produce more energy, but I've gone through the process of making this high energy molecule. I'm going to store it.
So can that molecule then react with the citrate synthase?
So it is what reacts with oxaloacetate to make citrate in citrate synthase. Okay. Once I've initiated that step, I'm pretty much committed to doing the Krebs cycle, and I'm going to end up producing more NADH. Right? So once I have that molecule, I might want to say, do I really want to commit to the Krebs cycle? If I commit to the Krebs cycle, I am producing energy. If I already have energy, I do not want to produce more energy. I need that molecule to go somewhere else.
But what's the trigger for the NADH to interact with
So the
citrate synthase?
The NADH will interact with the citrate synthase to stop that reaction. Because NADH is a high energy molecule. Well, the whole point of citrate synthase is to produce molecules that give us more energy. So NADH being high energy will come back and say, knock it off, citrate synthase, shut down. I want that acetyl-CoA going somewhere else. Okay. Instead of dumping it into the Krebs cycle, I'll dump it to do something else.
So will it bind to the citrate synthase and then cause that inhibition?
NADH, so that's your allosteric control. It's not going to be a covalent bond. It's going to somehow interact with it and change the structure, preventing either the acetyl-CoA or the oxaloacetate from binding. Kind of, sort of. So when we look at our big reactions here, which of those reactions do you think we want to flag as being really, really important? So our first one is important because we're starting with a high energy acetyl-CoA. That acetyl group can be used in a variety of other pathways. Okay, I think I'm doing this, anabolism, making molecules. Okay, we can use that to make lipids, okay, or our fats. Okay, we have a carbonyl group with a carbon on it. We could also use that to make amino acids. So if I have plenty of energy to survive, I'm going to take this now active acetyl group and I want to shuttle that somewhere else that can make something else for me. Okay? Build up walls and turrets so nobody can kill us. Okay? That kind of fun stuff.
So in that case, it skips the whole energy
It'll
cycle and it goes straight from acetyl-CoA to go make
Yep,
other stuff.
we'll ship that acetyl-CoA to something else. Okay? And we'll talk about that at the end of this chapter. Okay? What other steps might be kind of important?
The one where you make NADH.
Anything making an electron carrier, or NADH, is going to be important because our electron carriers can be used to make energy. So those are going to be important steps. We also have our FAD. Okay. Anything else in there stand out as like, ooh, that might be useful?
Making GTP.
What is GTP?
Precursor to ATP.
It's the same thing as ATP. ATP, sort of. ATP is adenine as our nucleotide. What is G? Guanine. It's still a high energy nucleotide carrying a phosphate group. Okay? So why do we use ATP as opposed to GTP? Go back to evolutionary biologists and ask them to explain why. I don't know. Okay? But GTP is still a very high energy particle, okay? Just like ATP. It can transfer phosphate groups. Very similarly, different structure, which means it's going to fit in different locations, but it's the same idea. Okay? So that is probably also going to be useful. That gets us direct energy. Okay, so we can star that differently. So we've got 4 of those giving us indirect energy. Okay. 1 of those giving us direct energy. And that very first one sends us to anabolism. Okay. So that was exciting. We used pretty much every single step except step 2 and step 7. Okay. So a lot of the reactions within Krebs cycle are pretty critically important, okay, for what's happening. Okay. The next part is if you look at all the enzymes, you're officially, again, responsible for those enzyme names. Okay. You might be able to pick out some of those. Anything involving NADH happened to be?
Okay, no.
That was ATP. A dehydrogenase. Your dehydrogenase is used NADH, okay? Because we're doing our electron carriers. That's what our dehydrogenase enzyme did. Okay. What was FAD, though? FAD is an electron carrier. So what kind of enzyme do you think we would use in an electron carrier system? Dehydrogenase. So we look at our reactions, a lot of those are dehydrogenases. Okay? The citrate synthase is the one that actually gets us into the Krebs cycle. We aren't actually in the Krebs cycle until we run that. Once we're in the Krebs cycle, we're doing a crap ton of dehydrogenase reactions, which means tons of energy is coming out. Okay? Another big one, GTP is our transfer of our phosphate. So we might go through and use a kinase. Notice it's not called a kinase. Oh well, doesn't always work. It's a synthase. What is it synthesizing?
GTP?
GTP is being produced in this case. This is a fun one. What's the name of that enzyme for step 5? Succinyl-CoA synthase, right? So what would that suggest it's making? Succinyl-CoA, right? What are we making in that step? Succinate. Where the heck is succinyl-CoA?
One of those ones where they figured it out in the opposite direction.
This is one of the ones where they figured it out in the opposite direction. Yeah. The succinyl-CoA in this case was the reactant. Okay. So recognizing some of those names, picking up with those patterns, and trying to see what's happening with them, good kind of general methods. So we ready to see some of the gory details? Yeah, that was an enthusiastic yes. That's what I wanted to hear. So here's my version of the citric acid cycle, or our Krebs cycle. It's the same thing as we saw with all those bright colors, except I removed all the colors and pretty much just drew the structures so that we could see what's happening within these. So if we start off, and I removed all the enzyme names, because I don't like enzyme names. We've got our pyruvate, which gets turned into our acetyl-CoA, and that was using our pyruvate dehydrogenase. I don't even feel like spelling them. Frick nuts. Okay. So once we have the acetyl-CoA, that can then lead into our actual Krebs cycle. Okay. To do that, we have to react the acetyl-CoA with oxaloacetate. So if we compare the structure of oxaloacetate to citrate, what changed? Oh, no, soup, sorry. Okay. If we go through and look at this, this is where it gets dicey on nailing this down. Our acetyl-CoA comes in with 2 carbons. Where is that acetyl group in citrate? Where are those 2 carbons?
Oh, third carboxylate up there.
So if we kind of go back to what I circled and then I very quickly backtracked on. What's the problem with circling that? I'm missing a carbon. So I could say, oh, okay, well then I meant to include this one.
That's already there, though.
But that's already part of it. So this is where recognizing the patterns on where your structures change becomes really important. Your acetyl group is actually coming in at the end.
Okay, then it just pushes the other ones.
So what will end up happening is that you will go through our whole enolate chemistry. That carbon will act as our nucleophile, and we'll go through and attack our carbonyl, which will break that open. We'll end up with, as kind of our intermediate, almost what's shown there, except instead of it being an oxygen out here, what do we have? S-CoA. What else do we need to initiate and finalize this reaction? And oxygen, if we go back to the previous slide, where are we getting that oxygen? Water is being used to cleave off our acetyl-CoA, or our CoA enzyme, and that gets us to our citrate. Right? So our pyruvate is coming in either on that side or it could be on the other side because it is symmetric across that. Everybody see that? Okay. So recognizing where it goes can help you try to predict what's going on with this. Questions?
Does the hydrogen just come from anywhere?
What hydrogen?
That alcohol?
Oh, I intentionally try to not draw hydrogens for that very reason. Where are hydrogens coming from? Yeah, we're in a water environment. We're in a protein. There's tons of acids and bases floating around. Protons will come on and off. That's why if you look at all the others, I don't.
Take care of that negative charge.
Yeah. That's why I didn't show any of the hydrogens there or charges on any of the oxygens because I was trying to avoid that very question. I did kind of need it for the very next step. If we look at that next step on citrase to cis iconitase, what happened? We did an elimination reaction. Okay, so we still have our pyruvate, but we've now lost the OH and an H. So we've done an elimination reaction in that next step to make our cis aconitase. Okay, why do we call it cis? Because the double bond is a cis double bond. That's its reference. Okay. What happens in the next step? We do an addition reaction. So in our elimination, we got rid of H2O. The very next step, we do an addition reaction where we add H2O. Okay, remember, we still have that hydrogen over there. So why bother to go through and do that? That seems kind of a bit silly. Okay, so if we think back to our previous reactions where we shifted that carbonyl, and you would ask astutely, why in the world would you do that? Okay, well, it's coming down to what process we're trying to do. We are trying to activate certain positions to make them more or less active. If we have the alcohol located in the middle there, we can't make a carbonyl. Why is a carbonyl useful? Carbonyls are good electrophiles. Okay? They could potentially react. They can also help polarize and shuttle electron density around. Okay? So what happens in our next step? If once we've got our D-isocitrate, what can we do? We can oxidize the OH, which gets us our carbonyl. Okay? So in that oxidation reaction, what does that mean? If we think we're doing an oxidation, we are increasing the oxygen bonds by decreasing our hydrogens. So we're losing the hydrogen now in purple. Where does that hydrogen go? NAD+, because it's getting ejected as H-. Okay? So that's getting picked up by our NAD+. So here's our first NAD plus coming into our reaction. Okay. We now have our alpha ketoglutarate. Does that structure look kind of familiar? Like if, for instance, I erased all that?
It's like pyruvate.
Looks like pyruvate. What could we do with pyruvate?
Start the cycle over again.
We could start the cycle over again. Let's back off of that a little bit. What did we do to pyruvate?
Added the CoA enzyme.
We did the CoA enzyme, activated the position. So if we looked at our product, we've brought our coenzyme A in there. So we're doing something very similar in this step as what we did with pyruvate. What else happened with that pyruvate? To get the CoA on, what else did we need to do? Begins with a T. Thiamine was involved. Thiamine's going to be involved in this case too. Okay, we're looking at this big dehydrogenase reaction again. Okay, what was the other big thing that happened in that?
The lipoic acid.
Yeah. Moving from pyruvate to acetyl-CoA, what changed? We got the CoA, yes.
Oh, we did
The decarboxylation. Look at the change from alpha ketoglutarate to succinyl-CoA. What happened? Whoops, sorry, maybe I should unerase. What changed?
We lost the CO2.
We lost the CO2. We are doing a decarboxylation in this reaction. Okay. Okay, so we've done our decarboxylation. We're going to expect the loss of CO2. Okay. And just like we did with our pyruvate, we're going to need all of those other pieces. We're going to need our thiamine. Okay, we're going to need our lipoic acid, or the equivalent of them. Okay, we're going to need our FAD, which will eventually turn into NADH. Okay.
Did we do that as well to get to that step, to get the alpha ketoglutarate? Looks like we lost.
We did not lose CO2. All we got rid of was H minus. So in the process of just getting rid of H minus in that previous step, we can shuttle that directly to NADH, or NAD. Okay, we didn't lose a CO2.
Where's that? So the deisocitrate, looks like there's a CO2 on the carbon where the H was. Is now not there on the.
Oh, snap, you're right. I completely didn't see that. You're absolutely right. So in the process of decarboxylating, or forming our carbonyl, we form our enol. The enol can be oxidized. Okay. Out to, well, it's not going to happen that way. We're going to have to oxidize first. Oxidize to our carbonyl, and then we can do the decarboxylation. Yeah, I completely did not see that. I don't know why I didn't see that. Good eyes. So we're losing CO2 there as well. Okay. This is our big process for cellular respiration. We're starting to kick out CO2s. Okay. Other questions on that?
Is that a decarboxylation too? Or
That ends up being a decarboxylation as well. Yeah. So we need all of those same big pieces. What happens in the alpha ketoglutarate? We just did that to our succinyl-CoA. Succinyl-CoA to succinate. What changed?
lose the CoA?
We just lose the CoA. Okay. Not a whole lot else is happening in there, so nothing's particularly obvious on what's happening there. I believe that's the one that gets us GTP. Might have to check me on that. Succinate to fumarate, what happened?
We lost some hydrogens.
We did an elimination reaction, and what we lost were actual just hydrogens. So we actually did oxidation reaction at the same time. Okay. Fumarate to malate.
Oxidation of the alcohol.
We didn't do an oxidation here. We actually just did an addition.
Water in it.
Just water. So yes, we see the oxygen coming on, but we have to realize when we're doing our oxidation, we traded sp2 carbons for an oxygen. Okay, sp2 is more electronegative than sp3. And our last step, malate to oxaloacetate. lose the water?
Maybe
Looks like it's just oxidized.
there's a
It's just an oxidation reaction.
From the structure, you built the structure on this though.
And the structure flipped on it. Why would the structure flip? I think it's because I initially picked the wrong end to be doing the pyruvate on, so it makes it look like the structure flipped. Okay. And it's likely because I just copied it from however it was drawn in the previous one. Okay. Kind of makes sense. So we're looking at the big structural changes. I want you to acknowledge those changes. Okay. I want you to overlay those on top of the big changes that are happening in here. So by looking at the structure, you can start to flag where you had oxidation reactions happening, where we were producing electron carriers. By looking at the structure, you could say this step is important because I'm doing oxidation reduction, and NADH needs to show up. Okay? So this is where acknowledging the structure or having some information about the structural changes or organic chemistry can help you predict what's happening. When it comes to the exam, what I will likely give you would be, let's just pick something here, this, and say, I don't know how many electron carriers are produced. You could look at the structures and say, well, I can't interpret structures. What else is given to you? The names. Okay? And I would probably even give you the enzyme name on this one. I don't remember what the enzyme name is, so I'm just going to leave it off. Okay, so that way, if you went through and memorized just the names of things and the enzymes, you'd still be able to answer the question. So I'm giving you two pathways to being able to get to the answer. Okay? Did you just memorize straight up names? Okay? Or can you look at the structure and interpret what's happening with the structure to say, yes, I think I need NADH.
What was the question?
In this case, do I need NADH or NAD plus in that reaction?
You're losing hydrogen somewhere, right?
You are losing hydrogen, but you are also losing oxygen.
Right.
So the net result is
But no.
nothing. When we look at our citrate to isocitrate, okay, nothing's happening. This is a bit weird because when you look at the reaction here, I just showed citrate to cisaconatase. Why are they not showing cisaconatase up here? Really what that enzyme is going all the way across. Okay.
It's like a pass through.
So it's a pass-through. So it's pulling the H and the OH off, and then it's saying, okay, exchange, back on.
It's really just kind of moving it
So it's just
down there.
the same enzyme is doing that process. Depending on what you want to look at, we may actually show the cisaconatase in there as an intermediate, okay? But it's not releasing that out to a new enzyme. That's a single enzymatic step. Okay? Questions about that? Okay. So the rest of these slides are pretty much looking at each of those individual steps and saying, let's break it down again and look at all the pieces. Okay. So our on-ramp is what they like to call it, taking oxaloacetate and converting it into citrates using our acetyl-CoA. Where did the acetyl-CoA come from? Pyruvate. Where did pyruvate come from? From glucose. How many pyruvates were produced from glucose? Which means when we run the Krebs cycle, we're running it twice for every glucose molecule. So we have to be careful on where we're looking at to see how much energy production is coming out. Typically, the question is, given one glucose, how much stuff do I get? Okay. But it could just as easily be switched to, given one pyruvate, how much do I get? So be careful in your frame of reference on what's happening within the citric acid cycle to make sure you're answering the question that's asked. Does that make sense? Glucose to pyruvate is known as our glycolysis. Pyruvate ultimately all the way through, or pyruvate all the way out and around back to oxaloacetate is known as our citric acid or Krebs cycle. I think people are trying to increase the use of citric acid as opposed to Krebs. I'm not sure of the history on that. Somebody must have got it.
The Krebs found it, and I guess he's getting older.
Yeah.
After he dies, they want
Yeah, so it sounds like Krebs pissed somebody off. That's really what that sounds like to me. Okay. So recognizing those pieces and kind of advancing them through. To do that reaction, okay, are we doing any oxidation or reduction? No. For citrate synthase? Okay, so we look at the structures, no oxidation or reduction, so we don't expect to see any electron carriers in there. Okay, we're just shuttling in the acetyl group into oxaloacetate. Okay. Notice that we are going to reform coenzyme A. So our coenzyme A acted as our good high energy carrier to facilitate and actually allow this reaction to occur, because the acetyl group is not a strong enough nucleophile on its own to attack the oxaloacetate. This is where that high energy sulfur bond comes into play and helps facilitate and carry this reaction across. Okay. We do have some big allosteric inhibitors here. We already talked about one, NADH. NADH eventually generates energy. Okay. This gets us into Krebs cycle. Once we're committed to our citric acid cycle, we are now producing energy. If I got enough energy, I don't want to do that. So NADH will come in and say, knock it off, don't start this. Okay. ATP is our end result of energy. It will also come back in and say, hey, stop this. There's already too much of me here. Okay. Succinyl-CoA will also contribute to this for alternate reasons. That's going to come back to looking at where succinyl-CoA shows up. Okay. Where is succinyl-CoA showing up? Why would succinyl-CoA shut down that reaction?
Is it the CoA part of it that does the actual blockage?
I would make the argument, what's produced in this step?
CO2,
And
NADH.
NADH is energy. What's produced in this step? Or NADH. Succinyl-CoA can double back and say, knock it off, because if you go through to make citrate, you're making more NADH. So stop it. Okay.
So it doesn't,
So-
the Krebs cycle wouldn't actually stop at that point, like this thing would continue on.
The
but
Krebs
that
cycle
NAD would,
pretty much once initiated is going to run. Okay, it's going to run out until those pieces can get siphoned off somewhere else. Okay. So we don't want those molecules floating around.
the NADH will just prevent further
Further
pyruvate
production.
creating,
Yeah, exactly right. Which then allows us to take that high energy acetyl-CoA and move that out to build something. Okay. This is producing energy so that we live. If we've got enough energy to live, now we can do something else with that energy. Okay. We can make cool cars. Kind of make sense? Step 2, our enzyme is a conatase. What is special about our isocitrate? It's a good fun question I didn't ask already. Everybody's favorite section in OChem 1. And potentially even our favorite section from carbohydrates.
it's like I love this one section.
So depending on your perspective, D was our biochem reference. The red dot in our structure here. In fact, actually,
Mention of
both red dots there.
the what
What is where? Yes.
Beyond that,
We have chiral atoms. Cis-aconitase we don't have to worry about. Citrate we don't have to worry about. There's no chiral atom. D-isocitrate, we have 2 chiral atoms. So we need to be specific in what form of isocitrate we made. Okay? Because it is an enzyme, it has a handedness to it, which means it only allows for one form to form. We're getting D-isocitrate. Okay? So that is pretty critical in realizing the enzyme is very specific in its reactivity. Okay? Any energy production? No. We're not seeing any oxidation reduction. There's no real phosphates being involved in this. We're pretty much clean all the way through. Okay. Isocitrate to alpha ketoglutarate. Don't worry about that mechanism to be discussed by classmates. That was from a previous class. And somebody was going to talk about it. We've got our enzyme, isocitrate dehydrogenase. How do we have a flag that it's a dehydrogenase?
The enzyme says.
Without the enzyme name, how do we know it's a dehydrogenase?
Oh, because NAD's involved.
How do we know NAD is involved?
Because you memorized that. No, I
Oh, fair enough. I'll accept that.
Oh, yeah, this is the hydrogen, this has to go somewhere.
This is our structural evaluation. If we look at that position, we've lost a hydrogen. Okay, we've increased the oxygen bonds, we've lost and decreased the hydrogen bonds. We've done an oxidation reaction. Okay, so that is our red flag that we're looking at NADH. That is our red flag that we're likely looking at dehydrogenase. Okay, this one is also doing the decarboxylation, which I completely didn't see. So thank you, Jimmy, for pointing out my failures. I appreciate that. That doesn't quite come out right, but I do appreciate that. Okay, and we end up forming our intermediate enol, okay, after the oxidation. Okay, this is going to have some allosteric effects. We're going to expect ATP and NADH to inhibit this. Why? It's making energy. Okay, so if we can avoid making more energy, we're going to try to avoid it. So we would expect some kind of effects in here.
And overall, every step that produces energy is going to be inhibited by more energy.
Pretty much anything that produces energy, yeah, is going to have some inhibition saying stop making energy. Okay, we are done. Sorry, I didn't quite see the clock because of the glare from my angle. So we'll pick up the rest of Krebs cycle on whatever it is, Thursday.